Everyone. So today I will be presenting an amazing article. Okay, the title is Inhibition of IL-11 signaling extend mammalian health span and lifespan. And this study published in Nature Journal on July 17, 2024 and explored the role of IL-11, interleukin 11, a pro-inflammatory cytokine in age-associated pathologies and lifespan. The authors examined whether inhibiting interleukin 11 can improve health span and lifespan in mice. The research was conducted by a collaborative team from various institutes, including the Duke NUS Medical School in Singapore, Bard Heart Center, London, also from the centers from the Australia and Germany and China. And the main investigators are the Aniza A, Widjaja and Stuart A. Cook. So this article is so important that one or two question can come in your competitive examination. Okay, because it is a very latest information and this must become. So start with the pathways. So five major signaling pathways that regulate lifespan across species. The first one is the ERK, extracellular signal regulated kinase. Second is the STK11, also called as the LKB1, liver kinase 1. Okay, B, liver kinase B1. And STK11 means serine threonine kinase 11. Third one is the AMPK, AMP activated protein kinase. Fourth one is the mTOR complex 1. mTOR stand for mechanistic target of rapamycin complex 1. And last pathway, which is a insulin like growth factor 1 and insulin modules. Okay, so these five pathways decide that you will age or not. So first, AMPK mTOR axis. So it plays a vital role in maintaining metabolic health and major role in aged organisms. Notable effect in the adipose tissue and the therapeutic inhibition of mTOR extend lifespan in mice. This is study suggestion. So now we will talk about the AMPK. AMPK or AMP activated protein kinase is activated in response to low energy level. Means when you take low calorie diet, then your AMPK pathway will be activated. It promotes catabolic pathway to generate ATP and inhibits anabolic pathways that consume ATP. Key functions of AMPK include enhancing glucose uptake, fatty acid oxidation, mitochondrial biogenesis, and autophagy, and also inhibits protein synthesis and lipogenesis. So, AMPK pathway is all about the energy conservation. Okay. Next pathway is the mTOR C1 pathway, which is a mechanistic target of rapamycin complex 1 and is activated by nutrient and energy abundance. So when you take the high calorie diet, so your mTOR pathway will be activated and it promotes the anabolic pathway which use the ATP. And you know that when you use ATP, heat production will occur, entropy will increase. So overall age, you will be become more aged. And it enhances the protein synthesis, lipid synthesis and cell growth, and it inhibits the autophagy. And autophagy is a useful because your old cell, damaged cells are destroyed, and from them body will get energy. So this will be inhibited, so it is lost for you. And the therapeutic inhibition of mTOR extend lifespan in the mice. This study suggests that the mTOR pathway inhibition can lead lifespan extension. And what aging studies tell that their primary focus on lifespan extension in yeast, worms, and fruit flies. Okay, and lifespan extension does not necessarily reflect the longer health span. Health span means disease-free survival. So that's it. And laboratory mice are suited for experiments due to their relevance to human aging pathologies. Now, chronic sterile inflammation. So, what is the chronic steriles? What is sterile? So, this is the acellular inflammation. Inflammation without cell due to the cytokines. Okay. So, it is a hallmark of aging. Aging associated with the dysfunction adaptive immune system characterized by immunosenescence and thymic involution, also associated with the inappropriate activation of the innate immune genes such as a gene encoding IL-6. Pro-inflammatory signaling factors like nuclear factor kappa beta and JAK-STAT3 are implicated in the aging process. The next we will talk about the IL-11, interleukin 11. So this diagram illustrates the signaling pathways involving the IL-11. Okay. So when IL-11 binds to its receptor, IL-11 receptor alpha, it interacts with glycoprotein 130 co-receptor, and this activation triggers two main downstream pathways. First one is the STAT3 and second one is the ERK, extracellular receptor kinase. And here you can see the STAT3 pathway induces the production of pro-inflammatory cytokines such as the IL-6, IL-8, IL-33 and CCL2, chemokine ligand 2. So all these will increase your inflammation. Okay. And ERK pathway activate the 19 kilodalton ribosomal protein S6 kinase. Okay, and this P90RSK will inhibit the LKB1. What is LKB1? LKB1 is nothing but STK11 pathway, and this pathway is responsible for activation of the AMPK pathway. So AMPK pathway is good for you, and this good pathway always inhibit the mTOR pathway, which is bad for you. Now this ERK and P90 RSK will inhibit this LKB1, so no activation of AMPK and now no inhibition of mTOR, and mTOR will produce the final product, which is the ribosomal protein S6, and this protein is responsible for the cell growth and anabolic pathways run. Okay, process. So ultimately your IL-11 will produce more protein synthesis, more inflammation, more lipid synthesis, and more entropy, and leading to the age. Okay. So what is the hypothesis of this study? The IL-11 may promote age-associated pathologies and reduce lifespan. So we will check. First, we will discuss about the methodology of this study. In this study, ethical standards for both human and animal studies were strictly followed. Now, cell culture. First, they done this cell culture. The primary human cardiac fibroblasts were obtained from the 52-year-old male and authenticated by the morphology and immunofluorescence for fibronectin and vimentin, and they cultured it. And why they cultured? Because it provides a human cellular model to study the effect of interleukin 11 and its inhibition on the cardiac fibroblast. And the second culture is the primary human hepatocytes, which is isolated from 22-week-old fetus, authenticated by the morphology and immunofluorescence for cytokeratin 18 and western blot for albumin. Okay, and this is used to study the role of IL-11 in the liver cells and its impact on hepatic function. So mouse models. So this study utilized 3 mouse models to investigate the role of IL-11 in aging. The first model, IL-11 receptor alpha deleted mice, you can see here, this mice has no IL-11 receptor alpha. So they have IL-11 in their serum but no receptor on the cell, so no effect of IL-11 will occur in these mice. Okay, was used to study the effect of IL-11 receptor deletion on the health span and the lifespan. A second model, IL-11 deleted or direct IL-11 gene knockdown is being done on these mice, and this allows researchers to directly assess the impact of IL-11 absence on the aging and related pathologies. The third model, IL-11 enhanced green fluorescent protein reporter mice were randomized to receive no treatment, then anti-interleukin 11, which is called as the X203 antibody, or IgG antibody, which is the placebo effect. Okay, this is for the placebo effect. These mice received 40 milligram per kilogram intraperitoneal injections every 3 weeks from 75 weeks of age for 25 weeks, stopping at the 100 weeks. This model provide comprehensive insight into the IL-11's role in aging and the potential therapeutic intervention. So if you have doubt about these mice, so they tagged every interleukin 11 gene with enhanced green fluorescent protein. Okay, so whenever the IL-11 will express in any tissue, it will reflect as a green color in the immunofluorescent image on the confocal microscopy. So who is this? This is the normal mice, also called as a wild type mice. Okay, so we will compare all these mice with these mice. So next step is the lifespan and health span study. So lifespan studies, mice were inspected daily and the age of death in weeks was recorded. And health span study. So now health span study, they conducted the glucose tolerance test, okay, then insulin tolerance test to know the better control of glucose, serum glucose, and blood glucose, and then echo MRI performed 1 day before glucose tolerance test, or euthanasia to assess body composition due to genetic modification or treatment. Then the frailty scoring, okay, used to evaluate the overall frailty of the mice as an additional measure of health span. Then they conducted the grip strength test. Okay, after that they conducted the wall body metabolic parameters like oxygen consumption, CO2 production, food intake, locomotor activity to evaluate the metabolic rate and energy expenditure. Then they done the molecular and cellular assays like seahorse assay. So in exam your questions like should like they will show these image on the screen, and they will ask what is this. Then there are 4 options. First one is the ELISA, so you will be mark ELISA because it look like ELISA, but it's not ELISA, it's a seahorse assay. So this is new information for you. Okay, and what is the use? So to assess the mitochondrial oxygen consumption rate and fatty acid oxidation. Okay, then next question, potential question, is this bomb calorimeter. So what is this? Then this is the IKAC 5003 control bomb calorimeter to determine the nutrient absorption efficiency. So someone can ask you that what is the what is the product which will be assessed in the bomb calorimeter. So it is the pickle matter, okay, the pickle matter of the mice, so nothing else, 0.9 gram of fecal matter of mice. Then the ELISA and colorimetric assay for the serum ALT, AST, these are the liver enzymes. Then the cholesterol, interleukin 6 triglycerides, also IL-6, IL-8 and IL-11 level in the cell culture media. We discussed about the fetus and cardiac myofibroblast, okay. And also we can measure the total collagen content in the mouse liver, gastrocnemius muscle and visceral gonadal white adipose tissue using the Widjaja total collagen assay kit, okay. So you can measure all this is the methodology only. So they conducted this much of test, and we will come to the result after this slide. So then they conducted the immunoblotting. So this is also new information for you and a very potential question for NEET PG, USMLE and FMG. Here immunoblotting is done with the help of STS page. This is electrophoresis, name of electrophoresis, okay. Sodium dodecyl sulfate polyacrylamide gel electrophoresis, okay, and followed by the specific antibodies. So this is the Western blot card, okay, and these are the proteins, all proteins you can see, and this is the age wise data, okay. This is 12 week and 110 week data. After that RT-qPCR done real time PCR, reverse transcriptase real time PCR. So here they extract the RNA from the mice, okay, mRNA, and they convert it into the complementary DNA, and after that they conducted the gene expression analysis study using the Continova Cybergreen PCR kit, okay. And by this method only they conducted the telomere length study and the mitochondrial DNA quantification study. And last methodology is the histopathology. So here histopathology they done HE stain they conducted the evaluation of the HE stain slides, and lipid droplet areas were quantified using the ImageJ software. Then they conducted the CD68 ISC was used to detect macrophages in tissue section, indicating the immune cell infiltration and the inflammation. What is ISC? Immunohistochemistry and immunofluorescence study, IF. So for various protein associated with the IL-11 pathway, such as the adiponectin, PDGFR, alpha, SLC10, A1, CD31, FHL1, SM22 alpha, and all these proteins will be studied on the single cell, single tissue by the co-localization of different protein in the same tissue. This all conducted in the confocal microscope, okay. I will show you image. And last is the data analysis. So RNA sequencing done with the library prepared with the TrueSeq stranded mRNA library prep kit. Then the sequenced on the high sequence or Nova sequence 6000 sequencer, and the data processed and analyzed for the differential gene expression using the DASEC2 and gene set enrichment analysis with the FGCA, okay. And statistical analysis, you know, the GraphPad Prism software, and using the appropriate statistical test. So now the final, which is the most important findings for your exams also and for your life also. So first finding of this study is the IL-11 is upregulated with age. So you can see here, this is the normal mice, also called as the wild type mice. So they euthanize these mice at the 12 week, then 25 week, then 50 week, then 75 week and 110 week. So there are multiple mice, okay, they have thousands of mice. So they euthanize some at the 12, some at 25, and some at the 110 week of age. So you can, and they assess the all these proteins on the western blotting from the liver, gastrocnemius, white adipose tissues. Okay, and they found that IL-11 protein, IL-11 is increasing with the age. Okay, this is a pro-inflammatory marker, and this is responsible for your age. Is it increased with age? Also, the pathway which is very beneficial for you, which makes you young, is this protein, phosphorylated AMPK, is decreasing by age. Okay, and also the bad pathway for you, which is the mTOR pathway, is these proteins are increased with age. Okay, and the byproduct of mTORs are also increasing with age. So here the data is also showing in the, this is the liver, and this data for the gastrocnemius muscle and white adipose tissue. So you can see at the at the 12 week there is only one, and here is the 9.5 IL-11. Okay, so you can see the confocal microscopic image of this. Also they can ask they will show you this picture and ask you that what is these photographs showing. Those, this is the confocal microscope with the 3 dyes. Okay, 3 antibodies. So you can see here the red one is the SLC10A1, and EGFP, I told you this is transgenic mice. Earlier I show you that third one. Here the every IL-11 gene which is activated is tagged with the EGFP dye, which is green in color. So if your IL-11 is very expressive, then it will show as the green color. But this is the young mice. Okay, IL-11 EGFP positive mice at the young age, 10 week, you cannot see any IL-11 expression at all. But at the 110 week you can see the heavy expression of the IL-11 at the 110 weeks. Okay, also similar finding you can see here in the white adipose tissue and the gastrocnemius tissue. Okay, gastrocnemius muscle. Okay, so it is a very increasing with the age. This is 10 week, so you cannot see the green color at all. But at the age of 110 week there is a high expression of the IL-11, and this column is for the wild type, which is the normal mice. So they don't have any tagged fluorescent protein on the IL-11 gene. So they can show this, but they don't have green color dye in it, so they are not expressing the green color. Okay, so finding number 2 is the IL-11 is associated with the senescence. Okay, so here they compared the this wild type and genetically modified, which is the IL-11 receptor alpha deleted mice. So they compared these two mice at the age of 10 week. So they euthanized both wild type and genetically modified at a 10 week, and also at the 110 weeks. Okay, so you can see the at a young age no difference in any protein. Okay, no difference at all. But here at the age of 110 week you can see the AMPK pathway, which is very beneficial for you, is all absent or just present in the wild type, this type of mice. Okay, and this AMPK pathway, which is useful, these proteins are useful from this pathway, and they are retained. You can see here at the 10 week of age and the 110 week of age, it is normal, and it is not even decreased. So it is good sign. So these genetically modified mice has good AMPK pathway proteins, and here the bad pathway, which is the mTOR pathway. So you can see here the mTOR pathway proteins are increased in the normal mice, compared to the genetically modified mice. Yes, there is mTOR proteins in the genetically modified mice, but it is very less from the normal mice. Okay, and the genetically byproduct of these mTOR pathway like P70S6, then ribosomal protein S6, P16, P21, all are increased in the normal mice compared to the genetically modified. So this mTOR pathway will not be very effective in the genetically modified mice compared to the normal mice. Okay, and same finding you can see in the white adipose tissue and the gastrocnemius. Third finding is the deletion of the interleukin 11 receptor alpha 1. Okay, improves metabolic mechanisms. So your metabolism will be improved if we delete IL-11 receptor alpha. So you can see the body weight. So at with age your body weight will be increased, your fat content will be increased, your protein content will be reduced. Okay, so here you can see this red is indicating the genetically modified mice, and this black color indicate the normal mice. Okay, so here the genetically modified mice showing the low body weight compared to the normal. Okay, and this is male female differentiation. Then this is the box and whisker plot, and the fat mass, you can see the fat is more in the normal mice compared to the genetically modified, but lean body mass, which is the muscles, are the increased in the genetically modified mice. Okay, so this finding you should not, that IL-11 is very related to the aging pathology. Okay, then you can see the liver triglyceride, which is increased in normal but not in the genetically modified mice. Also serum cholesterol and serum triglyceride, ALT, AST, all these liver markers, liver inflammation markers, are increased in the normal mice compared to the genetically modified mice. So this is the benefit, and this suggests the young, at a young age 10 week, and this is the at age of the 110 week. So okay, this is for the control. So you can see the at a young age both are same, but at an old age this level will be increased. Okay, and now the telomere length. So telomere length decide that when your cell will die, because at every replication telomere length will be decreased. Okay, but here you can see the telomere length is preserved in the genetically modified mice, and also the mitochondrial DNA copy number. Okay, so this is the biggest breakthrough. Now the finding number 4, deletion of the interleukin 11 improves metabolism and reduce the age associated pathologies. So here you can see same as the receptor deletion, same as the gene knockdown, IL-11, this time no IL-11 production. So finding is same as earlier, but here you can see the gene expression level. You can see the CCL2, CCL5, TNF alpha, IL-1 beta, then IL-6. All these are the gene associated with the production of the pro-inflammatory markers. And you can see the normal mice has overexpression, and the genetically modified mice has down-regulated all these genes. Okay, these genes are down-regulated in the genetically modified mice, so no inflammation at all. So this suggests the young mice, and this suggests the old mice. Okay, so here the forepaw strength, grip strength is increased in the genetically modified mice, and serum AST, ALT, all are the increased in the normal mice. And you can see the GTT and ITT, which is the glucose tolerance test and the insulin tolerance test. So these are very regulated very much regulated in the genetically modified mice. Okay. The next is the respiratory exchange ratio, which is very high in the genetically modified mice. Okay, and fecal calorimetry density shows no difference between two mice. They eat similar quantity of food, but grief strength is very much increased in the genetically modified, and fragility, which is a fatigueness. So easily fatigued. This normal mice will be easily fatigued by activity, but not this. Okay, genetically modified. So this is the breakthrough image. You will see this image on Twitter and on the internet, or if you are an academic person doing research in the lab. So this image you will see, definitely see in 1 to 2 months, because this is the biggest ever research done by the Stuart Cook. Okay, so here you can see the genetically modified mice at the age of 110 week, and this is the normal mice. You can see it is very fatty with the least lean mass and more fat content, and this is, and you can see the graying of their fur. Okay, and here this look like a young mice at a 10 week. Okay, so you can see that this mice is clearly aged compared to this. So this is the modified mice where they deleted the IL-11 gene. Okay, so next finding, they done the therapeutic inhibition, the main point of this article, and this will give you hope that your lifespan can expand if this research will successful in the humans. Okay, so therapeutic inhibition of the IL-11 improves metabolism and the lifespan. So here they inhibit the IL-11 by the anti-monoclonal antibody X209. Okay, this red color, and for the control they gave the immunoglobulin G intraperitoneally. So they started at the 75 week of age, so very late, and they took the response at after the 100 week of age, and you can see clearly that serum IL-11 and IL-6 level is decreased in the these cell lines treated with the antibodies. Okay, this P4, P7, P10, P14 all are the cell lines. We discussed the cell culture. So this P4 means progress for passage 4, passage 7, passage 10, passage 14. So this is the time, this subculture time. Okay, so P4 means they subcultured the cell line for 4 time. So it is very near to the original cell, but at the 14th time or culturing, it will very far from the cell origin. Okay, so you can see here the AMPK is preserved in the cell line treated with the IL-11 antibody, anti-IL-11 antibody. Okay, also the mTOR pathway will be reduced in the cell lines treated with the anti-IL-11 antibodies. Okay, and here you can see the telomere length preservation in the cell line treated with the antibodies, and also the mitochondrial DNA copy number is also preserved in the anti-IL-11 antibody treated cell lines, and same for the basal respiration is increased, and ATP production is also increased in the cell lines associated with the and treated with the anti-IL-11 antibody. So this is the heat map. This is the also most common question nowadays, and this shows the gene expression. So far red is showing that it is highly expressed, and the far blue violet color indicate that there is a low genetic expression. So here you can see the IgG treated, which is a placebo. IgG treated mice has very high expression of all inflammatory genes, okay, pro-inflammatory genes like CCL2, CCL5, IL-6. All these are the gene for inflammation, okay. All are increased, all are upregulated, but here you can see the pro-inflammatory genes are down regulated in the mice at the age of 100 week, which are treated with the anti-IL-11 antibody. Also you can see the cells with the fat adipose tissue. These are the adipose tissue which are very very tiny and small compared to the placebo treated mice, okay. And also you can see the inflammatory marker, which is CD68M1 macrophages, and here the inflammation is increased in the placebo treated, while the monoclonal antibody anti-IL-11 antibody treated mice show no inflammation at all, okay. And the final image, so this is the anti-IL-11 antibody treated mice at the old age. They look young. This is sign for the male, this is for the female, and you can see these mice has some ulcer on the skin, okay, but these are the perfectly fine mice and looks like young mice, okay. So this antibody is a miracle. And the finding number 6 is the just overall survival. You can see the overall survival of interleukin 11 knockdown mice are the more significantly more than the wild type mouse, which is normal mice. And also if we give IL-11 antibody to the normal mice, then their overall survival is also increased. So these points are very very important for your exam, and any MCQ question can come from this. And this article is also for research students, PhD students, to understand how to present the article and how to interpret the images and findings. So please share with your friends. And yes, discussion and the limitation of this study. So discussion: IL-11 upregulation observed progressive increase in the IL-11 expression in liver, white adipose tissue, and the skeletal muscle during aging, and activation of the ERK. You can see in this study we observed a progressive increase in IL-11 expression in the liver, visceral, adipose tissue, and the skeletal muscle during aging. This upregulation activates the ERKP90 RSK and mTOR P70S6K pathways inactivation, while the inactivation of the LKB1 AMPK pathway, forming a comprehensive IL-11 signaling module, okay. IL-11 is expressed in diverse cell types, indicating its widespread impact on the various tissue. And now genetic deletion benefits. Mice with the genetic deletion of IL-11 and its receptor IL-11 receptor alpha-1 exhibited several health benefits. These mice showed reduced inflammation, improved metabolic profiles, and lower marker of cellular senescence. Consequently, there were significant improvements in both health span and lifespan, underscoring the potential benefits of targeting IL-11 signaling. Okay. Therapeutic potential. So therapeutic intervention using the anti-IL-11 monoclonal antibody X2003 in aged mice resulted in enhanced metabolic flexibility, reduced fibrosis, and decreased the cancer incidence. This finding suggests that targeting IL-11 could be a promising strategy for extending health span and lifespan, offering a novel approach to mitigating age-related disease and improving overall health outcome. So limitation of this study, the mouse model constraints. This result observed in the mice may not fully replicate the complexity of human aging and disease. Therefore, the translational relevance to human remains uncertain. So, one of you will do this research on human. Then, scope of tissue analysis. Our study focused primarily on the liver, visceral, white adipose tissue, and the skeletal muscle. Other critical, this our study. I thought it's our study. Other critical tissues such as the brain and the cardiovascular system were not investigated, leading to a limited understanding of IL-11's role across all tissues. And biomarker and pathway focus, there was a heavy reliance on specific biomarkers and pathways in this study. This study may have led to omission of other significant aspects of aging that were not captured by their analysis. And the long-term effects and safety. So long-term effect and the safety of the chronic interleukin 11 antibody means inhibition in humans are not yet determined. Further research is needed to evaluate these aspects before considering clinical application. So thank you so much. Please subscribe my YouTube channel, and I will upload this type of informative videos from new articles, new information, and it will be very beneficial for you. So thank you so much. It's time to break.